Hey everyone, this is Yagnesh joining you live for our very first COVID Decoded live series. We're super excited to have you here with us for Raw Talk's inaugural live event, inaugural virtual live event. As you see, we're still working it out. But before we begin, we wish to acknowledge the land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and most recently, the Mississaugas of the Credit River. Today, this meeting place is still home to many Indigenous people from across Turtle Island, and we are grateful to have this opportunity to work on this land. Now, if you're anything like our team, over the last few months, you've probably been trying to wrap your head around the scale and the impact of the current global pandemic. But the rapid pace of COVID-19 discoveries and the mixed messaging when it comes to public health measures have made it hard to keep up. And as we adjust to this new normal, it's clear that the current pandemic has far-reaching effects on public health, mental health, marginalized and minority populations, medical research, and even our planet. So we decided to turn our annual live podcasting event, which was originally going to be focused on climate change, into a live stream series all about COVID-19. Over the next 6 weeks, we'll be talking about COVID-19 research and the impact of the pandemic in our everyday lives. To kick off the series, we'll start with the basics and understand what are coronaviruses and how does COVID, the virus that causes COVID-19, SARS-CoV-2, work. Before we begin and bring in our guests, we want to give a quick shout out to our Pillar Affinity sponsors, TD Insurance and MNBA. Visit affinity.utoronto.ca or click the link in the description below to check out the preferred insurance rates and credit card rewards for U of T alumni. Our guest today is Dr. Karen Mossman, a world-renowned virologist and, as of July, Vice President of Research at McMaster University. Welcome, Dr. Mossman. It's very great to have you joining us today.
Well, thanks so much for the invitation. I'm delighted to be talking to you today.
That's great. We'll just jump right into it. So can you tell us how you got started with virology? And what does being a molecular virologist entail?
So that's a tale from many, many years ago when I did my undergraduate degree at the University of Guelph. And I went to Guelph because I wanted to be a vet. It was my plan A, and I only had plan A. And I was fortunate enough when I was at Guelph to be able to volunteer as a first-year student in the large animal vet clinic, and I hated it. It wasn't what I thought, and I realized very quickly that that was not the profession I wanted to go into, and I did not have a plan B. Meanwhile, I was in a program, molecular biology and genetics, and one of my, I think it was either third or fourth year research projects or thesis projects was in a virology lab. And so I loved the research and I loved working with viruses. And as they say, the rest is history.
Yeah, it is. So what does being a molecular virologist entail? Like what's your day-to-day look like? Or I guess not as a PI, but when you are a molecular virologist.
So we're really interested in understanding how viruses work. And when we say molecular virologist, it's really at the molecular level. So, you know, how do viruses interact with their host cells? And I'm really interested in virus-host interactions. And so how do cells recognize viruses? How do they respond to viruses? And then, of course, how do the viruses counter-respond to allow them to continue to replicate, propagate? And it's this back and forth, it's the response, counter-response, counter-response, and it's that evolution between a virus and its host, which really has helped form the immune system, and also, the breadth and extent of viruses. So it's that I'm the most interested in.
Right, and as you said, there are so many viruses and millions that we probably even haven't sequenced or discovered yet. So can you explain the difference between what a coronavirus is and some of the other common viruses that we do encounter?
So there's, as you mentioned, a huge diversity of viruses, not just in their structure. So there's diversity in their structure as to what the source of their genetic material. Is it RNA? Is it DNA? Is it single-stranded? Is it double-stranded? Also the diversity in size, complexity. Some viruses code for as few as 5 or 6 proteins. Some viruses code for hundreds of proteins. There's diversity in the simplicity of their structure. Some are just the genetic material and a protein coat. Some have, they bring in proteins and nucleic acids from their host. They have a protein coat potentially and an envelope. So there's just huge diversity in structure, but also huge diversity in the types of mammals or insects or reptiles that they infect.
Right. So how is the coronavirus different? Specifically, how is SARS-CoV-2 different from what makes it novel? Let's just say that.
So the coronavirus family, again, is diverse. There's coronaviruses that infect many different types of species. There are 4 human coronaviruses that most people have been infected with. It's estimated up to 30% of the common cold, yearly common cold, are due to one of these 4 human coronaviruses. So what makes SARS-CoV-2 and the original SARS coronavirus, probably more interesting is that they didn't originate in humans. So, they were able to jump species into humans. So of course, as soon as you disrupt that virus host evolution, you see much more devastating effects. Whereas, many people will be infected with a human coronavirus that has been evolving with humans for many, many years, and it causes mild to no symptoms and common cold.
Right, and that's just because these have evolved with humans, and because of this evolution, they've sort of come to a balance between the infectivity and the lethality, and that's not the case for these viruses that jump species. Did I get that right?
That's absolutely correct. So, when a common virus has evolved in a human, not only does the human recognize the virus and know how to respond to that particular virus, the virus also knows how to respond, to the immune response of that particular host. So when all of a sudden now you have, a virus that has jumped species, the virus knows how to replicate because it has replicated in other species. But if the human immune system has never seen that virus, it hasn't generated the specific responses against that particular virus.
Right. So what makes a virus more likely or less likely to jump species?
So some of it is all about real estate, location. Does a particular virus that might be, found within a particular species, has it ever had the opportunity to even experience and potential to interact with a human or human cells? And then does it have the right mechanism, proteins to even get into human cells? When you think about a virus infection, we talk about 2 different things: susceptibility and permissivity. Susceptibility is, can it even bind and get into a cell type of a different species? And then once it gets in, is it able to, to replicate? And is that environment a suitable host? Does it have the right things that the virus needs within that particular host for it to replicate? And so that's when we talk about permissivity. So there's lots of viruses that we might be exposed to, but it can't get into our cells, or our cells won't support what they need. So we might be exposed to them, but they have no effect.
Yeah, most of the viruses by mass on Earth are probably bacteriophages, right? And they affect humans not at all.
Or you get into, the ocean and seawater, and it's estimated that per 1 milliliter of seawater, there can be 10 to the 8 to 10 to the 10 virus particles. when you swim in the ocean, you're exposed to those all the time, but they're a very different type of virus, and they don't get into or have access to, human cells. So it doesn't cause a problem.
Right. So you were recently awarded a CIHR grant to study the novel SARS virus. And actually, Arinjay Banerjee, a postdoc in your lab, was part of one of the first Canadian groups to successfully isolate SARS-CoV-2. What's the importance of being able to isolate and grow the virus in the lab?
So if you really want to understand the virus, you need to be able to work with the virus. We can learn some about the virus just by looking at clinical symptoms. But if you really want to know, how does the virus work? What cell types can it get into? How do human cells recognize the virus? What's the response of the human cells? You really have to be able to study that and ask that question. So you need the virus to be able to really to study it, to interrogate it. And then if you want to look for certain therapies, if you want to test new drugs, you need to be able to have a model system where you have the virus, so you can directly test the efficacy of a drug, to test the efficacy of a vaccine in an animal model. So it's really important to be able to have the virus to work with, to study, and to be able to, one of the first things, we did, and in the larger group that isolated the virus, was to make sure that colleagues across the country who have the right facilities also have access to the virus, because one group alone can't solve all the problems. Yeah. It has to be a collective effort, because there's too many questions to answer.
Right. We're still learning more about it, and I suspect we'll be learning more about it for years to come. So what are the aims of your grant, and what are the potential impacts of developing these infection models? I know you mentioned vaccine development earlier.
Yeah, so again, we're really interested at the level of really understanding the virus and the molecular virology aspects and how it interacts with its host. So we do, and we've already done, Arinjay's already, answered a number of the questions. We were really interested in, for example, what type of host cells or human cells can this virus even get into and replicate in? Obviously lung cells, because it's a respiratory tract infection, but clinically they've also seen gastrointestinal sequelae and other clinical signs. So it becomes really important to understand what are the different cell types, human cell types, that the virus can either enter into, replicate in, and spread from. And then how do those cells respond? Do they have a pro-inflammatory response? Because that, again, contributes to symptoms that we see clinically. Can the virus get into immune cells? Because as we know from HIV, viruses that can get into and potentially kill immune cells, that has huge implications for the immune response in the immune system. So we're really interested in, not just what cell types, but how do those cells respond? How does the virus counter respond? And, from that, we can help come up with some good model systems. So is there a good lung model system? Is there a good gut model system? So then, and if we can, get the model systems working, then we can work with colleagues who are very good at drug development. So drug development is not our expertise, but we have colleagues that are fantastic at drug development. But they need to be able to test their drugs or their combination of drugs in a model system. So that's where the collaboration ensues.
So what model system does your lab actually employ? I know Arinjay is a specialist in bat and understanding viruses in bats. What kind of model system do you work with in the lab?
So we've been working with a lung system, lung cells. We're just starting to, we have a new postdoctoral fellow in the lab that has expertise in gut and making organoids, so 3D culture systems. So again, that better recapitulates, what happens, these 3D organoid systems better recapitulate what happens in an organ in an individual. So we're setting up a number, even with human cells, of organoid systems. And we are interested in bats for a different reason, but it's thought that this virus evolved initially from bats, and we know that bats can harbor a whole variety of viruses and they don't get sick.
Right.
But yet they're very similar to us with their immune system. So we want to understand what are those subtle differences, and basically what do bats know that we don't know, and once we understand what those small changes are, how can we then use our modern technologies to really recreate the outcomes of those changes, so that we can use the same tricks that bats use as novel therapeutics.
Right, with gene engineering or novel drug discovery targeting specific immune types, those sort of approaches.
Absolutely.
And going back to modeling with organoids, and that's better than monolayers because we have more complexity of structures, or what about it makes it better?
Part of it is the complexity. When you have a single monolayer of cells in a tissue culture dish, that's providing the easiest possible advantage to the virus, because that's not how our bodies work. We know, for example, even in for respiratory viruses, there are mucus layers. So the virus has to get through a thick mucus layer. And then it has to get through, multiple cell layers to potentially get into the type of cells that it might replicate in. We know, for example, in lung for sure that the top layers are often being sloughed off. So, there are multiple with our intrinsic immune system, for example, mucus and all of this structurally, there's many, many more barriers. Whereas if you provide a nice monolayer, single monolayer of cells, no mucus layer, it makes it really easy for the virus, right? The virus is going to win because we're giving it all of the advantages. So, the more we can make it look like what would happen potentially within a human or within an in vivo model without having to work with an in vivo model, the more it makes it more challenging for us, but we can understand better than how does a virus get around some of these challenges.
Mm-hmm. That's very interesting. So going off of what you're talking about, bats, just leads me to think about some of the other pets. Most people seem to have some sort of contact with animals, pets or otherwise. Can they be affected? One, and two, can they be used? Can they be a source of transmission, like as a vector?
So it was really interesting. It must be now, 2 or 3 months ago, when the first report, I think it was a tiger in one of the zoos, had some symptoms, and they found that it was COVID positive. So, there have been sporadic instances where animals have been found to be positive. We certainly don't see the same sort of level of infection and spread, in household pets, dogs, cats, that, we've certainly seen in humans. Now, part of that could be, I don't know if we're testing. I mean, you don't.
That's true.
The more you look, the more you find, right? But there's certainly anecdotal evidence and reports of animals being positive. Now, how efficient that is, how efficiently they spread, a lot's not known. And it might be because we just haven't looked enough.
Right. Okay, that makes sense. Right now, I guess the priority is definitely trying to figure out public health consequences of the whole pandemic. So I guess we can talk more about how it affects humans now. So what do we know about what happens to the body once it's infected with the novel coronavirus?
So you've probably seen in the paper and heard there's a variety of different symptoms. So it's not sort of a one size fits all, and that's because not everybody's immune system is identical. And there's many things to consider. Why some might have an asymptomatic infection why others get extremely ill and sick. Part of it is initially how much virus were you actually exposed to? That's a huge component for any viral infection, not just SARS-CoV-2, but really for any viral infection. How much initial virus were you exposed to? If you passed someone who, sneezed and you walked past them a couple minutes later, you might have only been exposed to a very, very small number of virus particles. The chance is that is not enough to initiate an infection. Because again, you have all of these intrinsic, mucus layers, all these other
all the physical barriers that exist.
All the physical barriers. If you are, though, exposed to very high numbers of virus particles, just because of the nature of how you became infected, the virus takes hold because, your body, this is a new virus to humans, and if you've never been exposed to this virus before, in many cases for young, healthy adults, children that have an active immune system and a robust immune system, your innate immune system is there to deal with new pathogens that come in. Okay. That's what your innate immune system is for. And so, some people don't have any symptoms. Some might feel like they have a typical, and this is the problem with every year when someone has a cold. It could be any one of a number of viruses, right, because the response is the same. And so, you make some interferon, you make some cytokines, you feel lethargic, you might get a bit of a fever, you might have a bit of a cough and a sore throat. That's a normal response to a virus infection.
Right, and that's just the body responding to that, and the symptoms are a factor of the body's response.
Absolutely. And, all viruses, you'll get a slightly different response, but a general response is, making of interferon, which is a cytokine, making some pro-inflammatory cytokines, and collectively very similar types of symptoms. Mm-hmm. Now, of course, if you have a pre-existing condition so that your immune system is not working at full 100% capacity, often in the elderly, just over time, the strength of the immune system or the efficacy of the immune system wanes with age. That's where and not surprisingly, where we're seeing, the hardest hit in the elderly population. Right. Not only are their immune systems less robust, but by that point, there's often other, pre-existing health conditions as well. And so that's unfortunately where, we've seen the highest level of infections and severe clinical symptoms.
I see. So what's happening mechanistically in the body? Like, how does SARS, the novel virus, get into our cells and infect us, the pathogenicity of it?
So we know, we don't know everything about the mechanics behind it. We certainly know that one of the receptors is called ACE2. Right. So you've probably heard of, it's the same receptor that the original SARS also used, mm-hmm. Which, is not surprising because the two viruses are, I think, at the protein level about 95% similar. Wow. So we're not surprised that SARS-2 uses the same receptor as the original SARS. But it can't be as simple as that because, the symptoms and the ability to transmit are very different between the original SARS and this SARS. Mm-hmm. The original SARS was more of a lower respiratory tract infection, not an upper. The original SARS didn't transmit as efficiently, but it did have a higher mortality rate. Right. So in the original SARS, the overall mortality rate, I think globally, was around 10%. In Canada, it was 17%. Oh. I mean, fortunately, this virus does not have a mortality rate of 10 to 17%. That would be devastating. For sure. But this virus transmits much, much easier. And we don't understand all of the differences, but there are some things we do know about ACE2. We do know about another cellular protease that's involved also in how the virus gets in. We're starting to understand what cell types it replicates in and what some of the host responses are. We don't see as robust of an interferon response, for example, as we see with some other viruses. So we are starting to understand those subtle differences, which is now starting to allow us to understand, oh, that's why we see that clinically, or, oh, that's why it might be different from the original SARS.
Wow. Yeah, I mean, 5% difference, and we see so much difference phenotypically about how it presents in patients. It's incredible, and understanding it, like you said, is still an ongoing process. So, do we know about what any particular changes that we've already identified between the original SARS virus and the novel SARS virus that contribute to some of these different effects?
I think one of the biggest ones, one of the biggest differences is where not just ACE2, and we know ACE2 is one of the receptors, but it's not as simple as that. We know there have to be other receptors as well. But the original SARS really was a lower respiratory tract infection. Whereas when you have predominantly an upper and nasal sort of infection, that's partially what really lends itself to that transmission. Because any cough, any sneeze, runny nose, saliva, I mean, when you have a really upper respiratory tract and nasal type of infection, transmission is much more robust than if it's a really deep, lower respiratory tract infection. And another subtle difference with this virus is, and again, it could be due to those, if 5% changes, but 5% is still, quite a bit at the protein level. The ability to have the infections that are the asymptomatic infections. There were very few asymptomatic infections that we understood with the original SARS. If you really develop that lower respiratory tract infection, and almost everyone that had symptoms was positive.
Right.
Now we know there's a lot of asymptomatic carriers, and that enables spread. And I think that's really why, we're seeing such robust spread. If you don't know you have it, it's hard. It's easier to quarantine and to protect yourself if you know you're infected. If you don't know you're infected, it's so much easier to spread a virus.
Yeah, even if it might not affect you down the line, you're still acting as a vector to transmit to maybe multiple people down the line. So as you mentioned, the coronavirus research is still ongoing, and lots of groups internationally are collaborating with this. And I've seen your lab publish a lot of preprints about COVID. What do you think is the role of this international collaboration, and how the scientific community has been using preprints to inform the public, and some of the downsides and the positives of that?
Well, it's not just using preprints to inform the public, it's also to inform your colleagues. So some disciplines have used the concept of either preprints or, that concept of putting your work out into, if not a public forum, certainly the research community before an absolute publication. So there are some disciplines that have been doing this for years. Historically, in the medical field, biosciences, that has not been common. It's been very much get the paper published, and then once it's published, people can read about it. I mean, some people, even at conferences, at some large conferences, sort of in the biomedical field, will really only present research that's either been published or very close to publishing, because they're worried about getting scooped. And that's just, the nature of some fields. What's been really, I think it's been really gratifying, is, for one, this is now a pandemic that has affected everybody. I mean, even if you think about, other pandemics in our lifetime, there have been some regions that have been very hard hit. Other regions, Ebola, Africa has been very hard hit with Ebola. North America, Europe, No.
And that has to do
You
more
know,
with the environmental conditions of
it has,
replication.
there's a lot of different, yeah, absolutely, a lot of different conditions, how the virus spreads, where the virus comes from. This virus, we're all in this. It doesn't matter where you live. It doesn't matter. We're all in this. So that's almost given a camaraderie within the scientific community. And most people just want to get their work out there. And so preprints now have become, I think, standard. And that's good. I mean, it's good and bad. So it's good
You
because
need to
all of the
police that.
information is out there. But you have to be, it's like anything that you find on Google. Just because it's in a preprint,
That's true.
it has not yet been peer reviewed. Not to say that once it's peer reviewed, it's absolutely correct in the truth, because we're humans, and peer review means 3 or 4 people have reviewed it. Now usually they're experts in the field, but we're not perfect, we're human. So you do have to evaluate what you read in a preprint, maybe a bit more so. Again, you sort of get to know within any particular field who have very strong scientific records, publication records, are the methodologies that were used the right methodologies? There are ways that you can definitely evaluate. You do have to be careful, but I'm hoping that even after this whole pandemic subsides, that willingness to collaborate, to not worry about getting scooped, to make sure that the information is out for others to use, the sharing of resources. It's not just the preprints. Everybody is willing to share information, share resources, because we're all in this. We all want a vaccine. And honestly, I mean, if we're involved in helping the first vaccine or the first antiviral come out or not, I just want someone to do it.
Yeah.
Because that's going to make my life easier. So even if it's a completely different group and we've had no, we haven't helped at all, it doesn't matter. I mean, So I think there has been that collective, which has been amazing.
Right. So working with this very dangerous virus, I know everyone around the world is working on it. What kind of precautions do you need to take for working with something that's so dangerous and potential for a pandemic?
So if you're studying the virus the way we are, growing it up, looking at it with high titers, you have to do that in a containment level 3 facility. There is one in Toronto. That's where Aaron J worked with colleagues in Toronto at their containment level 3, because that's where the patient samples were.
Sorry, just jump in here. Can you explain what containment level 3 means?
So a containment level 3 laboratory. So there's four different what we call biosafety level or containment level laboratories. A lot of viruses that we work with normally are what we call level 2 or biosafety or containment level 2. We still work within a biological safety cabinet. So the virus or the pathogen or the bacteria or the cell line are still within a cabinet. So it's protected from getting contaminated, and the worker is protected from also being contaminated. Once you're in a containment level 3 facility, you wear much more PPE. So instead of just working within a biosafety cabinet, for example, the level of personal protective equipment is much more robust. All of the protocols are more robust. There's just more checks and balances in place. So that, the actual facility has to have negative pressure. So it's not just that you're protected because of the biosafety cabinet that you're working in. You're protected by the entire facility and the setup of the facility and the filtration of the air. So it's just that. And then, the highest level, which if you're working on Ebola and some of these other viruses, now you've probably seen, some of the movies, like almost like the space suits. Right. So there's the protection. You go in, so it's just increased levels of protection.
Sure. And that's what's stopping the spread from labs outside to the general public.
Absolutely.
So it feels like we're learning more and more about the virus at such a rapid pace. And we mentioned the role of preprints and how that works. And it can be hard to keep up with the latest information. And I think that poses a challenge for scientific communication. Like this is something that we've been trying to tangle with as science communicators, as graduate students. Um, there's been just so much confusion about in the general public about the origins, conspiracy theories. What are some of the misconceptions that you've heard of about the novel coronavirus?
Yeah, I mean, we've heard a lot. I think the data are pretty solid that it originated from a bat coronavirus. It was not. I don't think it was a
Yeah,
man-made,
I'm glad you
generated
tackled
virus.
that.
Yeah, no. I mean, now what we don't know is that in the wet markets in China, did it go directly from bats into humans? That's unlikely. I mean, the human coronavirus and the bat coronavirus are, I think, 96% identical, which sounds like a lot,
Yeah, but we just
but
learned about the 5% difference between the old SARS virus and the new one.
well, and that's about the same level of similarity between apes, gorillas, and humans,
Yeah,
right? So I mean, so it's 5% is a lot. So there's, and this is where, the folks that do evolutionary biology and, that becomes a fascinating area because, and, they're starting to look into, pangolins were thought to be maybe an intermediate host. Certainly with the original SARS, it was thought to go from bats to civet cats and then into humans. So I think a lot of that information has been, you can never say with 100% certainty, but the data are, are really heading that way. I mean, I certainly heard, originally because it was affecting lung function that, if you held your breath for a certain length of time, you could get rid of the, there, I mean, we've heard so many different, that it just, cell phone towers. I mean, in some
oh
areas of
yeah,
the world, they think cell phone towers. So they've burned and destroyed cell phone towers because there's a correlation between an area that has cell phone towers and a virus outbreak. So therefore it's coming, that's correlation. That's not causation.
for sure.
So yeah, we've heard lots of different interesting theories.
It must be frustrating as someone working on COVID to hear some of these conspiracy theories. But as our understanding of this virus and the disease quickly evolves, how can we as scientists better communicate the finding to the public and make sure some of these misconceptions are either resolved or don't arise in the first place?
Well, I think things like what you're doing right now is really important. Being able to have conversations, between, scientists and the public, being as open and honest that we don't have all the answers. But, here are the answers, or here's the information that we have. Here's how we interpret that. Again, the more, we're collaborative and the more you open up your data to, collaborators around the world, they can also evaluate that data. But it is important. It is important with the public to be as open and as honest. Sometimes data are what data are. People will hope for certain results, but the data are what data are. And sometimes we don't like what the data are suggesting. Right. Sometimes, we make a hypothesis and we're wrong, but that's the whole point of a hypothesis, right? You either prove it right or wrong. No one likes to be wrong. So I think, as long as you're doing science for the right reason and you have an open mind and you're open to all of the different interpretations of what your data might mean, and then you do have to have a way of being able to disseminate that to the public that makes sense to them. Mm-hmm. And it has to make sense to them, right? and then hopefully, everyone is, has their own opinion, but hopefully they are doing enough reading and informing themselves enough. But we're also human. Or I've even found over the 20 years of doing research that what we published wasn't necessarily wrong, but at the time we didn't have enough information or the right tools that we weren't able to. Now we have a different interpretation of what we saw 20 years ago, because now we have more pieces of the puzzle, or we have more sophisticated, methods to look at a question. And so now it's not that we did the experiment the wrong way or our data were wrong. We just interpreted the data the best that we could with the tools and knowledge we had. And now that we have more data, more tools, we realize, this is a different way to interpret. And that now actually makes more sense. So it's an evolving.
Right. It's a very iterative process, and you build on top of previous research. I think the point that gets lost, I feel like, when we're communicating to general public, is that we can be wrong, but the pursuit of science in trying to find new knowledge will eventually self-correct.
Absolutely. And the more people that work on it and look at it from all different areas, again, you're developing that picture. Every one scientist is one small piece of the puzzle. But the more people that do that, then all of a sudden the puzzle starts making sense. And even maybe if it's not filled in, you get a really good understanding of what is the final product going to look like.
Right. So I'm actually mindful of your time. This conversation has been very interesting. We've just flown past 45 minutes. So we're going to sort of go into the audience questions shortly. So if any of the viewers watching, please start sending in your questions. We already have some lined up, so we'll go and ask some of those. Let's see here. So one of the questions here is, I think we tackled it, but maybe you can just briefly expand on it. What exactly is happening when someone has the virus but isn't showing symptoms? And how does it affect how likely you are to pass it on?
So if you have what we call an asymptomatic infection, it's likely that you have some virus. It's replicating, it's shedding, but it's not either replicating to the extent that it could, or in the number of cells that it could, that the immune response isn't triggered to an extent where you're actually feeling the outcome. So you might be having a response, but not to the extent where, I have enough cytokines in my circulation that I'm feeling lethargic, or I have a fever, or, so you can have, either very, very mild symptoms, or, the symptoms just aren't sufficient enough that you recognize them, but you still, at a low level, could be shedding some virus.
Right, and that's to do with the tropism of the virus. And tropism, is the areas of the body or the cells that are more affected by the virus, or the virus can get easily into those cells. Right.
Or it's only a couple number of cells. It's a small number of cells, small number of virus particles that are being replicated. It's kind of like just flying under the radar.
Right.
So it's there, but it's flying under the radar, right?
But it's still there. You can still spread it. Yes, exactly.
But you can still spread it.
Exactly. Okay. I think that covers the main part of the question. Another question we have here, I think it's pretty interesting. Do we know about the mutational frequency of the genes in the novel coronavirus? And mutational frequency here would be the rate at which the virus has mutations in its genome.
So all viruses, when they replicate, mutate. I mean, even when our DNA replicates, right, there's a certain mutational rate, right? Which is why we get cancer and other diseases. And so this is an RNA virus. In general, RNA viruses tend to mutate, have a higher mutation rate than DNA viruses. But we don't, and they have been seeing mutations in this virus. And so one thing that's been happening around the world is a lot of sequencing so that they can start tracking, the genomics and the sequences of the virus, not just to see how it's, changing over time, but also within a certain geographical location.
I see.
There have been some mutations that they have seen. So, for example, one mutation that they have found around the globe, so in multiple geographical locations, is a mutation in the RNA polymerase. And it's a very specific mutation. And they have found that this alters the RNA polymerase and actually helps increase the mutation rate a small bit. Yeah. And so, again, if you have a mutation in a polymerase, the virus might be able to replicate a little bit better. If you have a mutation rate, for example, in the spike protein, that could affect how it binds to ACE2 or potentially other receptors that it could bind to, and therefore affect. I mean, we haven't seen a really high mutation rate, in part because coronaviruses also encode proofreading enzymes.
Oh, okay.
So, some viruses only have, a polymerase, and RNA-dependent RNA polymerases tend to have a higher error mutational rate than, say, DNA polymerases. But coronaviruses also express more proteins and have more genes than some other small RNA viruses, and one of them has a proofreading function. So we do see some, mutations, but certainly not on the same order as other viruses. Like HIV is a classic example. Yep. Extremely high mutation rate. Even within one individual, the virus is mutating and changing. And that's one of the reasons why, trying to find a vaccine has been really challenging for HIV because it mutates so quickly.
Right. And that mutation can just help it overcome some of the factors that identify in our body through the immune system. Right.
Absolutely. Yeah.
So since we've had air travel mostly suspended for the general public for over about 3 months now, do we know if there are already different strains on different continents because the world is more isolated than it was when the pandemic started?
They are starting to see, certain strains or clades, in different geographical regions. Now, that could in part be, as you said, because there's not this sort of one homogeneous, population because of all the travel. But we also know, again, from other viruses that, certain, there's also the genetics of the host. So, we've seen with other, populations where certain, populations are more or less susceptible to different types of viruses. And again, it comes down to that virus-host evolution. And depending on, particular, genetics within a particular population, right. Why we see, different susceptibilities and outcomes in, between, we see differences between men and women. We often see differences between different, ethnic backgrounds. So there's so many different factors that allow that to happen. It's not just one factor, but there definitely are, different strains or different clades that are more prominent in some areas than others.
I'm actually glad you brought up the ethnic differences. So in some of the countries that do have a diverse population, we've seen differences in outcomes for patients of different ethnicities. Do we know if that's related to genetic factors or is that something else?
That's where it becomes, I don't think we know enough yet if it's strictly genetics. But there's so many different things, and this is why for not just a viral infection, but a lot of different diseases, you always try to understand what are the underlying factors. Right. In some cases, it's genetic. In some cases, it's environmental. In some case, there's all diet. There's so many different, and it's usually multifactorial. So it's rare that you can look at, an infection or a disease and say it's due to one factor. It's usually due to a multiplicity of factors.
That makes sense. I don't think there is enough data out there to make a conclusive statement about that, but it is something that is very odd. I haven't seen this before for some of the other viruses that we've encountered. Let me just look at some of the questions that we've got coming in. I see one of these questions. I think we've sort of tackled it, but if you could briefly expand on it. How do we know that this coronavirus came from bats or another particular species?
So for one, bats are really interesting. They harbor, many, many, many different types of viruses and virus families. And there are actually papers from, I think, even before the original SARS coronavirus broke in 2003, where they had started sequencing and identified, different viruses in different bat species. There are some papers even before the original SARS outbreak that had found coronaviruses in bats that, had very high, like in the 90% similarity to some of the human coronaviruses. And so, and this, certainly happened not just with SARS, but some of the other Ebola virus, Marburg virus, again, sequences found in bats that have, and this is historic. So, there's even a Egyptian tomb bat. So again, from thousands of years, and they were able to get enough to sequence some viral sequence from an Egyptian tomb bat. And in one of the proteins, I think it was the polymerase, that fragment, and it was only a fragment, but that fragment that they sequenced was 100% identical.
Wow.
To, so again, we know bats have harbored, coronaviruses and bats have been, for millions and millions of years. They're one of the most diverse mammalian species known. So they have probably harbored these viruses for longer than Homo sapiens have even been, on the planet. So again, good evidence that that's the origin of where these viruses came from.
It's very interesting. And you mentioned bats have been harboring these viruses for a long time. What about the bat immune system makes it possible to sort of be a reservoir for these viruses, but then not show any pathogenicity or symptoms because of it?
So again, part of it, I think, is because, bats are so, as a species, or the order, I think there's something like 1,400 different species of bats. They've been interacting with viruses for millions and millions and millions of years. So they've learned how to deal with viruses for, again, longer than Homo sapiens have even been on the planet. And so they developed some tricks. So we're starting to learn what some of those evolutionary, tricks are. So, for example, bats don't show signs of disease because they don't, to some viruses, make a pro-inflammatory cytokine response. And Aaron J., as part of his PhD thesis when he was in the University of Saskatchewan, found an element, a negative regulatory element that's upstream of pro-inflammatory cytokines. Wow. And so, and he was able to prove that by now if he takes that element and he puts it in front of a human pro-inflammatory cytokine, virus infection does not make that pro-inflammatory cytokine. And if he takes that sequence away from bats, now they make pro-inflammatory cytokines. So we're starting to learn what some of those really small, subtle differences are.
So the pro-inflammatory cytokines are the ones that are causing this, the presentation of the symptoms in any of the animals that are infected. Am I getting that right?
Yeah. So a lot of, a lot of times, people don't necessarily succumb to the virus per se. They succumb to it's the same thing with influenza, like the Spanish influenza. You hear about cytokine storm,
Right.
So it's an overactive immune response, and it's usually the pro-inflammatory cytokines, and then they have, negative sequela if you have an overactive pro-inflammatory cytokine response. You want some inflammation, but some, right? It has to be controlled. So, and so, bats don't show signs of disease because they don't have, they're not making the pro-inflammatory cytokines.
But they can still be diseased then if they don't have a inflammatory response, but still harbor the virus. Do you classify them as having the disease or not?
Well, it depends on what your definition of. So they have the virus. They don't have the disease.
Oh, that's true. I guess it is the.
Yep.
All right. So we have very little time left, and I think this will be our final question. So just to wrap up, for the proteins that are encoded by SARS-CoV-2, how many do we know the function of out of those genes or proteins? And how do we go about that?
So I think because, they're coronaviruses and between the family of coronaviruses, there's certainly, a lot of similarity. So we know a lot about the core proteins, polymerase, spike protein, some of the ones that form, but then it's all of these accessory proteins. So, not required for the absolute replication, not required for the physical structure. Mm-hmm. And we're just starting to learn, what do some of these sort of accessory proteins, what do they do and how do they modify, the immune response? So that's work that our lab is doing that. Other labs are doing that. So we know some about the virus, but it's those little extra bits that you really need to understand.
Well, actually, just I know we said the final question, but if you can just entertain me for one more, what are some of the pressing questions that we still need to answer about the novel coronavirus?
So I think one of the most important ones is if you've been infected, and even if you have made an antibody response, are you protected? I think that's one of the most important ones, because we've seen evidence of people that have been positive and then they've been negative and then they're positive again. Wow. So we don't know, is that just, again, in the negative test, is it because they flew under the radar and the test just came out negative? So, did they have an infection all along and they just didn't clear it? But that's still over a long period of time. Or were they infected, they cleared the infection and then got a mutated or different strain of the virus. So we still don't know. And this becomes important when we get the serology testing working, just because you're seropositive. So it says, yes, you have been infected. You have antibodies. We don't know yet, are those antibodies protective?
Right.
And that is going to become, that to me, at least from the clinical, that's the most important question.
Right, and that also has implications for vaccine development, because if our antibodies can't recognize it, then vaccines might not work for it. Well,
absolutely. Or in a vaccine strategy, are you making the right type of antibody? You're making an immune response, but is it the right type of immune response?
Very interesting. So thank you very much, Dr. Mossman, for being on this show, and it's been very nice and informative to have you on, and I'm sure our listeners also agree. Where can our audience get in touch or keep up with your research?
So we do have a lab website. So if you just Google Mossman Lab, McMaster.
I think we have it linked in the description too.
Yeah, we do have a lab website. I mean, most of my guys are busy doing all this work that I don't know who's actually maintaining it right now, but we have been trying to put all of our findings into the preprints as well. So if you just look and do the Google or the PubMed searches, that's where the latest information is going to be.
All right. Great. Thank you so much for coming on. So coming up next Thursday at 3 p.m., join Raw Talk host Jesse as he sits down with Dr. Sharmistha Mishra and Dr. Linwei Wang to talk about the epidemiology and the modeling of COVID-19. For the full COVID Decoded series schedule, please check the description box below. We'd love to hear what you think. Please fill in our feedback survey, also linked below. And until next time, keep it raw.